Hello and welcome to this webinar titled PDA Technical Report on Low Endotoxin Recovery Implications to the Industry, hosted by Biopharma Asia magazine and presented by Friedrich von Wintzingerode, Senior Manager of Global Analytics Science and Technology and Microbiology at Global QC at Roche. My name is Stephen Edwards and I'll be your moderator. Now please allow me to introduce our presenter. Friedrich studied biology with a focus on microbiology at the Technical University of Braunschweig, Germany. Diploma at German Strain Culture Collection, DSMZ, Braunschweig, Germany. Degree at Institute of Microbiology and Hygiene, Charité, Berlin, Germany. Since 2001, employed at Roche Diagnostics Pharma Technical Operations, working as micro lab manager on various topics, environmental monitoring, cleaning analytics, microbiological IPC, and analytics for release. In his role as Senior Manager Microbiology, Friedrich led several Roche/Genentech micro-expert teams, for example, identification and microbiology testing. He is the head of Roche/Genentech's Global Endotoxin Expert Group since 2013, and Roche/Genentech Global SME on microbial product contaminations. I'll now be handing over to our presenter. Welcome, Dr. Wintzingerode.
Yeah, thank you very much for the nice introduction. Welcome, everybody. Today I would like to talk about a new PDA Technical Report, number 82, on low endotoxin recovery, and yeah, especially about the implications to the industry. So maybe to give you some background about the LDR topic in general. So the LDR topic, or low endotoxin recovery, was first reported by Chen and Vinther in 2013, and it has been broadly evaluated in certain matrices, which are commonly used for biologics formulations and certain therapeutic proteins. So it was initially found in a formulation which was citrate and polysorbate or phosphate and polysorbate. These were the initial findings. And because of the LDR effect, a pharmaceutical product contaminated with endotoxins may go undetected by the compendial BET testing or LAL testing, according to the harmonized USP, EP, and JP checkers. So this was the basic finding. When we apply the BET per compendial guidance, there's the possibility for certain biologics formulations that LDR occurs and that we underestimate the true endotoxin contamination. Because of this concern, the US FDA CDER began asking companies to conduct endotoxin spike and hold recovery studies to determine whether a given biological product shows LDR. The LDR effect is something one can only detect if the spike, undiluted spike product, is held for a certain amount of time. So it's not detected during routine method suitability testing of the compendial BET. So because of this request, which followed, I guess, 1 year after the initial publication of the LDR finding, firms have conducted numerous spike and hold recovery studies, and the results, were not really convincing. In many cases, a great variability was seen, and sometimes even contradictory results. And that's most likely because some products are more likely to exhibit LDR, and also very important differences in how these LDR studies were conducted. There are a lot of things which need to be considered, and which can cause variability, which is not necessarily related to any true biological findings. And this was simply the setting when CDER began asking to conduct these studies. I have to mention that these studies were only requested for BLAs and are still only required for BLAs. Because of these, it's a complicated situation that the studies were not really clear and the outcomes were not in all cases convincing. There is definitely a mutual interest between industry and FDA to work together and to develop a science-based and data-driven strategy in dealing with the LAR phenomenon. That's really key, that this concept, how to deal with the LAR phenomenon, is done in a science-based and data-driven strategy. At a very early day, there was a lot of debate and discussion, and not in all cases data-driven, but a lot of, let's say, discussions which should have been in a different way, not too much data-driven. So therefore, the PDA Biotechnology Advisory Board sanctioned a so-called LDR task force in early 2015, 2 years after the LDR finding was published at a PDA Congress. And the task force was composed of subject matter experts from academia, FDA, the biopharma industry, and reagent supplier and testing companies. So it was a very diverse team, and really covering all the people involved in this topic. So just here a few names. I don't want to go into detail, but just to give you an overview who was involved. So my colleague Dayu Chen from Eli Lilly and me, we were co-chairing this team. So we had Julie from Merck, Alan from Lonza, Joseph Hypergenics, Gregory from bioMérieux. bioMérieux was involved because of the Hyglos company, which they recently bought. Hyglos is a vendor specialized in endotoxin testing. We had Jennifer from ACC, well-known LAL provider. We had Jessica and Patricia from FDA, Stefan Novartis, Chris from Bristol Myers Squibb, Jack Levin, he was our academia partner. He was the one who invented the LAL assay. So he brought in depth knowledge about the LAL assay, and having him on board was absolutely helpful. We had Jean from Amgen, Nat from Pfizer, Cheryl started, but then later on retired. Julie took over. We had Johannes from Microcode, Dean Genzyme, Anders from Novo Nordisk, Cass from Charles River, and Rini from Biogen. And last but not least, Josh Eden from PDA, who was facilitating our meetings and was a great project manager. So this was a big team, and it took us many meetings, face-to-face meetings and CCons to come up with this technical report. And I would like to give you an overview and also some details about this technical report in the next slide. So the key sections are given here, and I'm focusing on these key sections really because I think these are the sections which have the biggest impact on industry. The report also includes an overview about endotoxin detection, endotoxin structure, a history of endotoxin standards. I can definitely recommend reading these sections because it's very difficult to pull out these information from other sources, and many of the information there was very unique for this report. These are the sections which have the biggest direct impact on the industry. So first, and one may not think about this as a very important topic, but it was definitely a very important topic, the glossary. We defined key terms like endotoxin LPS. There was a long discussion for many years whether endotoxin and LPS are the same or are different. We could agree on a common definition of this, definition of the CSE, LER, masking, NOEs, all these terms which were used frequently in the debate and the discussion. They are all defined here, and I strongly recommend to read this section and to adopt these definitions. I think it's very important to understand the topic and to make the LDR life easier. Then we have a section about LDR hold time studies. This was one of the key sections in terms of providing guidance for setting up these studies. It's a points to consider like section, so it's not a cookbook, but it provides the most important things which are needed for planning and conducting scientifically sound and harmonized LDR hold time studies. And this is really one of the key sections in terms of having something for the lab to conduct studies so that variability is reduced. There's also a section about the proposed mechanism of LDR. That's something which was based on recent publications and the input from the different team members. So we differentiate between LDR and the BET test interference. That's also a very important aspect. And there is a proposed two-step model of LDR given in the tech report. But of course, like every other model, this model will change if more and more data will be published. So I think that's relatively clear. A very important section is also about the potential safety impact of LDR and the LDR mitigation strategies. So mitigations via sample treatment are described, and the evaluation via biological systems. So this is especially important if LDR has been observed and companies need guidance what to do in such a case and how to maneuver through the BLA and to meet FDA requirements. Last but not least, there's an appendix section where a total of 12 different case studies from the different companies are given. And I think that's also a very helpful source of information to see what kind of outcomes are possible, and also the broad spectrum of different LDR effects. Yeah, so some more specifics about the glossary, and I would like to start with the control standard endotoxin. So the definition we agreed on was endotoxin preparations other than the international or national reference standards that are traceable in their calibration to the International Endotoxin Reference Standard. This is what we call a CSE. The CSE is secondary or tertiary and standard, and it's commonly purified from E. coli. And it's usually manufactured and certified by a LAL reagent manufacturer for use with a specific lot of reagent under defined assay conditions. So it's important to emphasize that a CSE in principle could also be generated from non-E. coli LPS, and in principle could also be generated in-house, meaning at a certain biopharma company. It doesn't necessarily need to be produced by a LAL vendor. But in most cases, this is done. In most cases, it is E. coli, and in most cases, it is done by a certain reagent vendor. And I think it's also very important to use these type of CSEs. But that's simply the definition. And it's important to keep this in mind that in most cases, it is E. coli. And my personal recommendation is to always check whether the LPS type has been changed. Most likely this will not happen, but it's not put in stone that it's always E. coli. But I think it's important for every biopharma company to keep an eye on this, because of course this would introduce potential variation if a certain vendor would, for example, change the LPS type. The second definition is about endotoxin, and this took us more than a year to agree on this definition, but it was really one of the key definitions. Some of you might be aware of the term LLR versus LER. So there were some authors which proposed to coin the term LLR because of the difference between LPS and endotoxin. So people saying that LDR is not really an endotoxin effect, but only for LPS, because LPS would be the purified form and endotoxin would be the natural form. We could clarify that this separation or differentiation doesn't make any sense. Endotoxin and LPS are synonymous terms. So the major constituent of the outer membrane of gram-negative bacteria is composed of lipid A, the core polysaccharide, and the O-antigen polysaccharide. Endotoxin is also known as LPS, lipopolysaccharide. And that's given in an overwhelming set of publications. It's really important to not differentiate between these terms. All the terms LPS, lipopolysaccharide, and endotoxin can be used interchangeably. and it's really important to keep this in mind. So the term LER, or low endotoxin recovery, is defined as the inability to recover greater than 50% activity over time when endotoxin is added to an undiluted product. LER cannot be overcome by dilution. And that's a very short and simple definition, but every word here is important. So it's really, for example, the term over time, which refers to the whole time which is needed to detect such an LER, and also very important, undiluted product, meaning that's really the difference between the classical BET method suitability testing, where people can also spike the undiluted product. This is not possible if one wants to detect LER, and this does not, spiking the diluted product does not mimic a natural contamination. So the term masking, this is what we define masking as a type of interference that may result in low endotoxin recovery. The term is not new. It has been used since 1998. There was a publication talking about protein masking. We simplified this here, and yeah, it's simply an interference, a certain type of interference, which may result in an LER. Also very popular is the term naturally occurring endotoxin, NOE, and this we defined as endotoxin prepared from gram-negative bacteria produced under defined conditions and with minimal non-chemical processing, e.g. centrifugation and filtration. So the minimal non-chemical processing, that's an important part here, because a CUE is usually purified with a chemical protocol. For an NOE, one does not use any chemical step like phenol extraction, for example. So now coming to the LER whole time study, and here it's really important to start with a very fundamental definition. So LER whole time studies are not part of Compendial Endotoxin method suitability testing. That's really very, very important. So we do not consider these studies as part of the Compendial testing, but they are defined as supplemental studies, and they are mandatory for LER detection. So keep this in mind whenever you want to set up these studies. It is important for your quality system. Don't mix it up with method suitability testing, which is purely Compendial testing. And without a carefully designed LER whole time study protocol, the results can be both confounding and contradictory. So it's very important to carefully design the LER studies, and I will come to the key and pain points later on. Otherwise, the results are not clear, and you have to deal with these results because you usually generate these data under GMP. So the technical report provides points to consider for planning and conducting scientifically sound and harmonized LER whole time studies. Alternative approaches may be used if adequately justified. That's also very important. It's not like FDA wants you to use only these points to consider. As long as one can justify an alternative approach scientifically, one can assume that FDA will accept this, but the points to consider given here are definitely strongly recommended, and I will come to some aspects where the FDA representatives in the team made it very clear that these are things which they definitely do want to see during whole time studies. Another very important aspect is the differentiation between LER studies and so-called QC sample whole time studies. So the validation of a QC sample storage time, which one can define as the maximum allowable time between sampling and testing, is not in scope of the technical report, and it should be conducted separately. And there are various talks and presentations out there which combine these LER studies with whole time studies, and there was a lot of confusion in the network about what is the difference between these different studies, and isn't this the same? We decided, let's keep these things separately. It makes life much easier. And here are the reasons why we came up with this. So LDR whole time studies should determine whether a product has LDR under conditions which are relevant for the manufacturing process, not the sampling and testing process. That's very important, and I will come to this later. QC sample hold time refers to all sample types, and it is a general GMP requirement, so it has nothing to do with LDR. So validating a QC sample hold time is something which FDA frequently requests during inspections, and which is simply the basic GMP requirement, independent from LAL, independent from endotoxin testing. It's something everybody has to have. The storage of a QC sample can occur under conditions which are very different from manufacturing conditions. For example, one can freeze samples, which is usually not possible for certain biologics. But on the other hand, many of the principles described in the technical report may also be applied to the validation of the QC sample storage time. So it's not like they are technically completely different, but they are simply two different GMP entities. Now some aspects about the test method and parameters. So it's very important to use the same BET method for LDR whole time studies, which is or to use the same BET method for whole time studies as one for use for routine testing. So don't use, for example, a KTA assay for an LDR study if your routine testing is done with a KTA, for example. And in addition to this, the vendor of the lysate and the endotoxin standard should be the same. The procedure for sample and lysate preparation should be the same. The data analysis method, for example, the respective algorithm used for generating a standard curve should be the same, and whenever possible, the sample dilution. If the validated dilution from a product-specific method suitability testing fails to produce a valid result in an LDR whole time study, it is possible to use a higher dilution, which is not exceeding the MW, because this is something we have seen quite frequently, that, for example, if one has validated a 1:50 dilution in the method suitability testing, this validated dilution does not necessarily work in an LDR whole time study. And people have to increase the dilution to, let's say, 1:75, for example. And this would be still okay if it's below the MVD. The spiking of the undiluted sample, again, that's basically what we are doing. With the LDR whole time study, we always spike the undiluted sample. So it is important to know that FDA gave us the feedback during the tech report writing that LDR whole time studies should only be done with EP samples. Yeah, there were FDA, before we wrote the technical report, in some cases also requested LDR studies for drug substance and IPC, but the very starting point from now on is CC samples only. And just in case of an LDR confirmed finding, additional process steps might be requested to be tested. But the initial screening is only focused on DP. And if the DP is lyophilized, it is recommended to reconstitute the product following the product-specific instructions whenever this is possible. Again, spiking must be performed on the undiluted sample, that's clear. And the endotoxin source for spiking are CSE or RSE. These are the preferred standards. And NOEs may be used as a supporting study. And this is something which has also been discussed very intensively in the past. And I have to emphasize here, the term preferred standard means the initial screening should be done with CSE or RSE. And it's not possible to see screening with NOEs only. And it's also not possible to do a screening with CSE or RSE, and in case of an LDR effect, simply switch to an NOE, which might not show LDR, and then close the case and say, okay, I saw an LDR with CSE or RSE, but since I haven't seen LDR with NOEs, there's no true LDR effect. This is not longer possible. This has been tried by a couple of companies during BLA. FDA was very clear on this from very early on. such an approach is not possible. So the volume of the spike solution should not exceed 10%. That's important to not over-dilute the undiluted sample. Needs to be considered when setting up the study. The spike level, also very important. The endotoxin level after the validated sample dilution should be near to the midpoint of the standard curve. And the specification limit of, for endotoxin should be taken into consideration. So keep all these two parameters in mind and look at your standard curve and then decide on the appropriate endotoxin level. The level of spike endotoxin shouldn't be too high because LDR is also concentration dependent. So for example, spiking a product with 500 EU of 500 EU per ml is most likely not meaningful. Hold time study setup. A few more words about the different approaches which are possible. So in general, a multi-aliquot approach is recommended to avoid sampling repeatedly from the single container. There were initial studies or early studies reporting spiking of a certain solution and then repeatedly taking samples from this spike solution over time. But we do not recommend this. We definitely recommend the multi-aliquot approach. So the first approach would be about sample spike and time point samples aliquoted, stored appropriately, and assayed at the specified time point. And this is given in the figure below. We have the spiking at T0. We store the different, then we aliquot and store the different aliquots for a certain amount of time. In this case here, 4 hours, 8 hours, up to 7 days, and pull out one of these aliquots per time point and test this. There's also a so-called reverse time fashion mode possible, which many companies prefer. And in such a case, the day 7 samples are spiked first, and day 0 samples are spiked last, and all time point samples are assayed at the same time. So both approaches are equally well suited for LDR studies. Some words about storage containers and batch requirements. So the storage containers were under big discussion for many years. And this is simply because of different findings using different storage containers. PDA again made it very clear the only requirement they have is that LDR studies should be conducted using containers which are appropriate for endotoxin studies, meaning no adsorption, no interference. So there's no longer the requirement to conduct these studies in the original container, because this has been initially requested by FDA that people have to conduct the studies in the original container. In some cases, when studies were requested for drug substance, people conducted studies in stainless steel containers, which of course can cause absorption effects of the spike endotoxin to the surface of the container. And to avoid this variability, FDA agreed on this very simple definition. So meaning, in principle, all LDR studies can be done in appropriate sample containers which are provided by the different vendors. And this really simplifies the overall process. Regarding the batch requirements, it is recommended to use samples from 3 batches to ensure that the variability among processes is captured. But each company and each lab should determine the appropriate number of batches to use for validation depending on the batch size and the formulation. So this 3 batch requirement is not put in stone, but I think it's simply what most of the companies are using. Sorry for this texty slide here. I recommend to read this after the webinar. What is given here are two aspects about storage time and temperature. The very initial studies, or many companies conducted studies at a highly standardized time and temperature. Many companies use 7 days as a maximum hold time and 2 to 8 degrees for storage temperature. When looking at the different data provided by the different task force members, FDA was pretty clear they want to have the study done at the process relevant temperature and time. And the term process relevant refers to the process steps that are most likely to have an impact on LDR. For example, the addition of polysorbate in the respective chelator, like citrate, for example. And that means that process relevant in many cases is simply room temperature and not 2 to 8 degrees. The process relevant steps can be categorized into the following groups. So if the process relevant steps are conducted at refrigerated temperature, studies should be conducted at 2 to 8 degrees for 7 days. So meaning if your relevant step, the step where you add the polysorbate, for example, is conducted at refrigerated temperature, and this can be, in your specific case, a temperature range of, let's say, 4 to 15 degrees, just to give a number. In such a case, you can store your sample in a classical standard switch, which gives you 2 to 8 degrees. Because nobody wants you to mimic exactly your process conditions in the lab. If the process relevant step is conducted at room temperature, the study should be conducted at 20 to 25 for the process relevant time. Meaning, if your process step, relevant process step, the addition of polysorbate occurs at room temperature, and again, room temperature can be, for example, 15 to 35 degrees, you can conduct your study in a classical micro incubator. And this again was very important to standardize because nobody can mimic, or it would be very challenging for a micro lab or endotoxin lab to mimic 15 to 35 degrees Celsius in a lab. So that's why we agreed on, okay, let's take a classical micro incubator at 20 to 25. And the process relevant time, meaning if your process relevant time is 7 days, then use 7 days. If it's only 3 days, use 3 days. And if there's a process relevant step conducted at 2 to 8 degrees and one or more process steps performed at room temperature, then the study should be conducted at 20 to 25 for the respective relevant time. Meaning 20 to 25, room temperature is the worst case. This is the temperature you should use for your study. If the whole time studies generate inconclusive results, or if additional information is needed, the whole time study may be extended by adding more time points. And just a note here, the tech report does include a detailed example for risk-based evaluation to determine LDR whole time study temperature and time parameters. So there are examples given in the tech report how to define the exact temperature and time for a given setting. Some words about the data analysis and interpretation. So to determine if LDR is present, the endotoxin recovery is calculated by comparing the amount of endotoxin in the sample against the amount of endotoxin in the spiked LRW, meaning LAL, reagent water reference sample. Again, the tech report describes exceptions where the theoretical endotoxin value can be used, but this is the general setting you compare against your water control. And a minimum of 4 time points must be conducted to ensure valid and accurate results. So meaning that a whole time study where you only measure T0 and T, let's say after 7 days, is not sufficient. You need to have at least 4 time points for an accurate result. And whenever there are 2 consecutive data points below 50% recovery, you have LDR. So this can be testing after 1 day and after 2 days, below 50. If the recovery goes up again after a longer incubation time, it doesn't matter, you do have LDR. In many cases, the recovery drops down after a certain storage time and it stays below 50%, but it is also possible to have this below 50% in the middle of the time curve. We have seen this. If the last time point results in a recovery of less than 50%, additional time points should be tested to aid in the analysis of LDR masking and to ensure that the LDR result is accurate. That's very important. If you have a 4 time point study and the very last, only the very last, shows below 50%, you cannot call this LDR. You need additional time points. because you need to have these 2 consecutive data points. It's really important, 2 consecutive, not 2 independent, no, consecutive. The data from all batches must be plotted and interpreted separately. So there's no averaging of the mean allowed. That's really important. If you test 2 different batches, you have to assess them separately. And each of the batch must fulfill the acceptance criteria. If any of these batches has a confirmed failure, meaning less than 50% is recovered for 2 consecutive time points, the product or process step is considered to be exhibiting interference with the recovery of endotoxin activity, meaning if 2 of your batches are fine, but 1 batch shows LDR, your entire process step shows LDR. It's also very important to emphasize that the time 0 endotoxin value, meaning immediately after spiking, should not be used as a reference because as LDR may occur within minutes, and in such a case, referring to the T0 value would lead to highly biased reference. So therefore, T0 should be measured, but the recovery should never be calculated against T0. So now a few words about the proposed mechanism of LDR. First of all, it's very important to differentiate between LDR and the BET test interference. So the LAL or BET test interference can be caused by, for example, suboptimal pH, unbalanced cation concentration, unspecific enzyme reactions, and so on. So there are different possibilities. And as these interferences are often concentration dependent, usually dilution of the sample is the best approach to overcome this interference. There are publications from the early 90s showing that dilution can overcome more than 90% of the inhibition. And in such a case, you can achieve a valid PPC recovery. In contrast, LDR is a time-dependent, dilution-independent lack of recovery, and valid PPCs are typically achieved. Meaning, if you observe LDR, you usually have valid PPC recoveries because you dilute your sample for the PPC, but you spike the undiluted sample for the LDR finding. So these are 2 different things. Of course, there are certain cases where the effects can overlap, but in principle, these are 2 different effects. And this is important to emphasize because, again, with the classical MST, method suitability test, you cannot detect LDR because you don't have this whole time component. You can't spike diluted samples with the MST. So the proposed 2-step model of LDR is based on a publication from 2017, and I don't want to go into too much into details because again, new published data might change this model. But basically, what is happening is that endotoxins occur as in micellar structures, so they, at least in an aqueous system, they do not occur as single molecules, and they are connected to each other via divalent cations, in many cases magnesium, for example. And if this is illustrated by the black dots here. So they connect the different endotoxin molecules with each other and form these micelles. If you now add a chelator, then these divalent cations are somehow pulled out of this endotoxin micelle, destabilizing the connections between the different LPS molecules. And again, then if you, in addition, but this effect would be reversible by simply adding more magnesium. But if you then also have surfactants present, then a new structure is formed, and these LPS or endotoxin molecules, they let's say coagulate with the surfactant, and the divalent cations are captured by the chelator. So what we see here is a structural rearrangement, and that's something which is again very different from inhibition, where the endotoxin structure is most likely not affected. So the classical, what we call classical LDR, meaning LDR caused by certain buffer formulations, citrate and polysorbate, or phosphate and polysorbate, this is a structural rearrangement. There are other types of LDRs possible, like protein masking, where most likely the endotoxin is attached to a certain protein, and this would be a different model. So this here describes the formulation-driven LDR. And the proposed two-step reaction mechanism is confined to those LPS fractions with particular salt bridges, and this is what I mentioned with the divalent cation. In other cases, the function of the divalent cation is replaced by charged substituents. And not going too much into details about the chemical structure of LPS, but LPS is not a uniform molecule. It does have highly conserved structures, but it also has parts which are highly variable, and this further complicates the entire LDR topic. And I have to say that the the LPS complexity has been totally underestimated in the past 30 years of endotoxin testing in the biopharmaceutical industry. When I started doing endotoxin testing around about 20 years ago, this was a very simple, or it was introduced as a very simple, easy to do QC test, and nearly everybody can do this. But this was a very bad introduction, and it was deadly wrong, because the LAL testing is one of the most complex testing principles, because endotoxins are so complex and they have so many different structures. And what is given here, if the connection between the different LPS molecules is not given by divalent cations, but by charged substituents, then the model which I've discussed here before doesn't apply, and then the entire LPS micelle is not affected by LDR. So consequently, the presence of complex forming agents may not destabilize the LPS interactions for those LPS preparations, and complex forming agents like citrates are less likely to reduce the rigidity of LPS aggregates, meaning that certain endotoxin types are more tuned to LDR compared to others. And this is something we have to consider, and this was one of the main reasons why FDA wanted us to use an LPS type which is tuned to LDR. It doesn't make sense to conduct studies with an LPS which can never show LDR in any case because simply of its chemical structure. The safety impact was also a topic which was heavily discussed within the team, but also in at many congresses and workshops. So the LDR suggests that pyrogenic products could be distributed to commercial use in patients. Yeah? So looking at the findings, FDA was really concerned about product release to the market which would include or which contain undetected endotoxins. So the team reviewed peer-reviewed scientific literature, public data on recalls and adverse events, and all available copies of the Centers for Disease Control, Morbidity, and Mortality weekly reports. And all these sources did not reveal any instance where a BET failed to detect pyrogenic levels of endotoxin in samples of products released to the market. So that's the good news. It really looks like there's no historical finding where LDR has caused release of a pyrogenic product to the market, which then caused any adverse event to patients. Meaning that published historic data provides no direct evidence for BT failing to detect pyrogenic products. So this is the good news. However, and that's really important to consider, due to the complexity of biological drug formats and materials, historical data do not necessarily provide an adequate basis for safety assessment of new drugs, supporting the need for LDR studies in BLAs for specified biologics regulated by CJ. So it is good news that our current molecules on the market are safe, but this doesn't mean that new products are also automatically safe. We have new drug formats, we have new formulations. It's very hard to compare the new products with the old historical data set. And that's why FDA is requesting this type of studies, these LDR studies, for new products going to the market. It is not requested for products which are already on the market. That's not the case. It's only for new products going to commercialize to the commercial market. Now talking about strategies in case one has observed LDR. And this is, from a technical perspective, a very complex topic because there's no one solution fits all possible. But of course, one needs to consider that many findings can be resolved by simply following the points to consider outlined in the LDR whole time study. So many so-called LDR findings were simply false positive or negative, however you want to call this, simply because of poor study design. It's very easy to generate a reduced recovery simply because of, for example, lack of appropriate vortexing or simply the study setup. So whenever one has an LDR finding, it's very important to confirm whether this is a true LDR finding or just poor study design or study execution. It's also important to understand the potential influences of the product itself in combination with the formulation, buffers, and temperature and alcohol recovery. And this is something usually the companies can only do by themselves because only the companies have the in-depth knowledge about their products. And that's why contract labs and service providers are limited, because usually biopharma companies do not provide this level of detail about their products to the contract labs. So in many cases, meaningful LDR troubleshooting is not possible without in-depth knowledge about the molecule. It is recommended that the following approaches be performed in sequence so that the most practical and straightforward to the potential solutions are studied first. If this approach is not successful, the combination of the individual strategies might be considered. And all potential solutions to LDR are empirical. It's really, that's what we have to admit, that in most cases it is empirical. There's no single treatment known to work for any specific category of products or formulations. Again, there's no one-fit-all solution possible. And coming back to the sequence, it's really important to start with the easiest troubleshooting aspect or the easiest solutions. So meaning it doesn't make any sense to start working on a very sophisticated sample treatment if one, for example, can simply fix the by switching to a different vendor. So start with the simple things, and if this doesn't work, go to the more complex. So evaluation of the product. So again, there are different LDR effects. It can be driven by the formulation buffer, but it can also be driven by protein masking. Protein masking might be may be considered in cases where the protein is substantially hydrophobic or electropositive, meaning that it does have certain regions of positive charge that can bind to the negatively charged endotoxin. So in case you have a confirmed LDR finding, in case you can exclude poor study design or poor study execution, talk to your product specialist and ask them about potential binding sites of your negatively charged LPS. It's really helpful, and we have seen such an example recently here with Roche. It's very important to address this LDR topic as a cross-functional effort. It's not, you cannot work or you cannot solve LDR issues if you only work within your silo, only working within the endotoxin lab, or only within micro. You have to talk to your product specialist. The isoelectric point of a protein is not a reliable indicator of LDR. It has been proposed as an indicator of LDR, but it has shown to be not very helpful because all proteins are sputerionic. The tech report suggests studies to separate the effects of product from formulation components. So meaning, for example, in case you have a certain formulation buffer without the protein, conducted spiking study with a formulation buffer, and you can easily see whether the buffer causes LDR or not, and whether it can contribute to the LDR effect. Using proteinase K has shown in some cases to improve the recovery of spike LPS. There is a publication from 1998 showing that POD-K can be used for endotoxin masking, but it does not work in all cases. So one has to simply try for the POD-K to digest the protein and release the endotoxin. Another possibility is addition of dispersants to the sample. There are different brands available, like Pyrospores, the EndoRS kit from Biomarrier, or the BD100 buffer from Charles River. And all these components have shown to resolve issues related to PPC recovery, and in some cases they have also shown to work quite well for LDR issues. But it's not a general solution, and it's again always case by case. The composition of these components is proprietary, so we don't know. We mean biopharma companies. We don't know how these solutions are set up, but they are simple to use and to evaluate, and it's simply recommended to use different combinations, different volumes, times, temperature, mixing protocols, and again, it's a pretty easy and straightforward approach. Another possibility, and it worked in some cases, is addition of divalent cations, for example, an excess of magnesium, and lowering the pH in the sample containing citrate to weaken the chelating effect could resolve LDR in some cases, and but based on the pKa of the functional groups, phosphate buffers would not be expected to benefit by lowering pH. So this is something which would only work for citrate. But I have to say, addition of magnesium chloride can also be lead to false positive results, meaning mimic good recovery, but simply based on enhancement of the LAL without being related to endotoxin. So be careful and have the respective controls that addition of excess divalent cations does not lead to false positive results. Organic solvents have also been proposed as a potential solution, or substrates like bovine, serum, albumin. But again, there's no general solution. One can try these additives, and it might work. Be careful with DSA. Use a nanotoxin-free preparation, because in principle it can contain high loads of endotoxin, and then it would mimic or generate false positive results. Also very important, and whenever possible should be used as a starting point, simply try different LAL reagents. It's not always possible, because in certain company settings the release testing is fixed. It needs to be a certain vendor, it needs to be a certain method, because the method, for example, has been filed to the agencies. But if you have the variability and have the flexibility to use a different vendor and a different method, it's definitely worth trying, because we have seen some cases where simply switching to another method and vendor solves the issue. A very last step in the mitigation or evaluation of an LDR is if you have confirmed LDR finding, if you have done the very best to overcome LDR by all these mitigation strategies, and you still cannot overcome LDR, you still have to deal with your inefficient or not sufficient BT testing, then it is important to go the next step, and this is to determine if the spike endotoxin is detected in a biological system. And this biological system is usually the rapid Hygiene test. So when the endotoxin spike product samples are found to be pyrogenic, the manufacturers should implement the rapid Hygiene test as an interim QC release test until a suitable in vitro test method can be developed and implemented. And the development and implementation of a valid in vitro endotoxin test can occur after BLA approval as a post-marketing commitment. Meaning, if you go to the BLA, you don't have a solution for your endotoxin detection method. It's still affected by LDR. You do the spiking studies with the rapid Hygiene testing, and if it's still pyrogenic and your rabbits show the fever signal, then implement the rapid Hygiene testing as an interim, and that's really important, interim QC release test. And this is something which companies should consider as being a bit more complex, which have filings or products in different markets, meaning US and EMA, Japan. Because the rapid Hygiene testing is something which is more complicated under EMA regulation, for example, because of animal protection rights. When endotoxin spiked or health products are not found to be pyrogenic, meaning that LDR masking leads to a non-pyrogenic LPS, pyrogen tests for product release may not be required. The firms are asked to conduct additional in-process endotoxin tests prior to the addition of the formulation component, which is causing LDR. Endotoxin testing of input raw materials should be in place, and usually tighten microbial control of all subsequent processing steps. Meaning, improve your overall microbial and endotoxin control. And if your product is not pyrogenic after spiking and holding, then this LDR effect does not have the consequence that you have to use rapid Hygiene test. You can somehow live with this deficiency, and FDA will accept your DNA without conducting rapid Hygiene testing for that release. Some general thoughts at the end. So the technical report provides guidance and support based on the current knowledge about LDR. That's really important. It is a snapshot. It's not the ultimate knowledge book about LDR. It's simply based on the available knowledge during writing. The PR is not a cookbook, but it provides points to consider for the industry to develop science-specific approaches which address health authority expectations. Currently, the health authority expectations are only from FDA seniors. I'm pretty sure that other health authorities like EMA and Japan, PMDA will adopt these expectations, because LDR is a general safety concern. It's not related to certain agencies. Yeah? So I personally expect other health authorities to take over these principles, which have been outlined by FDA and which are captured here in the technical report. The TR focuses primarily on sterile protein drug products. A risk-based approach is recommended to assess applicability to small molecule injectables, meaning that if your company is producing small molecule injectables, short peptides, or everything which we would not consider as a biologic, they do not fall under CEDAR, but since, again, this is a safety concern and not only a compliance concern, one should conduct a risk-based approach to see whether LDR studies are needed, even if not requested by a certain health authority. That's simply the recommendation. There are still, even after working on this technical report for a couple of years, there are still open questions. And the topic of special interest are, for example, the development of LDR demasking methods. So we definitely need more data, more publications about demasking protocols to help the industry to develop these protocols for other products. We still don't know exactly what are the relationships between different endotoxin types and different LDR types. So, for example, which endotoxin types are prone to matrix-driven and API-driven? We simply don't know. We lack a lot of knowledge here. and still not finally address this LDR mask protein, endotoxin still pyrogenic or biologically active. We still need more spiking studies, published, peer-reviewed spiking studies to have a final answer on this, and we might see differences between the different LDR effects. But we have to have more data about the biological impact. Firms are encouraged to generate more LDR data and publish them in peer-reviewed journals. It is not sufficient to publish these data in congresses, to give talks and slide decks. We need to have peer-reviewed journal data. Otherwise, the data are not really meaningful. And if people are doing this, this will increase our database and will allow adjusting the LDR guidance over time. That's really I think it's very important to emphasize. I know that's very critical for many biopharmaceutical industries, companies to publish in peer-reviewed journals because of IP reasons, but it is very important, and otherwise we cannot move on, and otherwise we have to live with the status quo. That's the end of my presentation. Thank you. Stephen, you want to take over? Stephen, are you online?
Our first question is the PDA TR is saying that either CSE or RSE can be used for LER whole time studies. Which one is preferred?
Yeah, so this is a difficult question. Indeed, the TR is not preferring one of these two different endotoxin types, and gives some flexibilities. I think it's a very difficult to answer question. We as a company have decided to use both analytes because we have seen conflicting results in the past, meaning LER occurs with TSE but not with RSE and vice versa. And we found it very difficult to choose the appropriate analyte. So we as a company decided to use both and simply deal with the results. In case they are contradicting, we have a certain approach. If they confirm each other, then of course things are easier. But from my perspective, one cannot really recommend one over the other. I think there are certain topics related to this. One aspect is the availability. RSE is more expensive. The number of batches is somehow limited. The handling is a bit more complicated compared to CSE. That's definitely on the con side. The CSE does show variability. It's always batch related. So using CSE only would or could lead to some variability over time. So therefore, we as Roche Genentech decided to use both. But it's really hard to say one is better than the other.
Thank you. Our next question is, there are studies and papers showing that LER is limited to CSE/RSE and NOEs do not show LER. Why does the TR recommend CSE/RSEs for LER screening?
Yeah, that's a very good question, and we had long discussions about this topic. So I think there are two components. First of all, these early data could not be confirmed with later studies, meaning that there are also published data out now showing that even NOEs can show LER. So it's not a simple equation. CSE and RSE do show LER, and NOEs do not show LER. This was simply a very small data set, and new data do not confirm this initial finding. This was one aspect. The other aspect was knowing the large diversity of endotoxin types, knowing the large variability. FDA did not agree to use a certain NOE preparation where nobody really knows about the whether this is representative for different companies for different settings. And another concern by the agency was, what about the stability of these NOEs? So if, for example, you prepare an NOE, how stable is it over time? And the agency was very concerned about introducing more variability if people would conduct studies with NOEs. NOEs are simply poorly defined. It can be a tap water preparation, for example, having different species and different LPS molecules, but it can also be generated from a pure culture. And to eliminate this variability and to use a worst case analyte, the FDA insisted of using one of the pre-established standards. And again, new data published beginning of this year show that it's not this simple equation. Unfortunately, LER is not limited to highly purified endotoxins.
Thank you. I think, unfortunately, that's all the time for questions we have. But thank you for your answers, and thank you, audience members, for your questions. Before I finish the webinar, I would like to ask our presenter, Friedrich, if you have any closing remarks.
I think, yeah, if there are further questions, would be nice to see all these questions. I'm happy to answer these, and I think LER is nothing which has been resolved by the TR. It's simply we have more guidance here. It's important to have a standard approach so that all the pharmaceutical companies approach the topic in a similar way. It will help us to streamline our filings. But again, it's not the end of all research, and it might be even seen as the start of more focused research on this topic.
Thank you. We may have time for one more question, so let's go for, what about MAT testing for pyrogenic samples instead of RPT?
Yeah, that's a very good question. So MAT is definitely a promising tool. In some cases, it could be shown that it's not affected by LDR. The only problem with MAT instead of RPT is simply FDA does not accept MAT as a replacement of RPT. That's simply why the agency currently wants us to do rapid pathogen testing instead of MAT. That's the main reason. There are different or separate initiatives working on this. MAT could replace RPT, but that's a long way. And yeah, that's simply the main reason. And another thing might be, and that's why I think FDA is not completely wrong, it's still not completely understood whether MAT is really or capable of replacing a biological system. So MAT is an in vitro assay, and there are some data sets lacking which demonstrate comparability between RPT and MAT. And that's the main reason why FDA does not accept MAT as it is now.
So thank you all once again. I would like to take this opportunity to thank our presenter once more. That was Friedrich von Wintzingerode, Senior Manager of Global Analytical Science and Technology and Microbiology, Global QC at Roche, for sharing his knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com. We'll get those answers back to you. Thank you, everyone. Have a great day.